Okay, before we move on a little bit more here to the cell, a couple things I should probably also hit on. When we were talking about aerobic versus anaerobic metabolism, the product of catabolism in order to create energy is generally pyruvic acid as part of the process. In an anaerobic state, that pyruvic acid is not easily broken down, and it becomes lactic acid, and that lactic acid obviously increases the acid within the body. Not to mention is basically a toxin to the body. When we have aerobic metabolism, that pyruvic acid then is able to be broken down into carbon dioxide and water, which is obviously easily eliminated from the body. So through the process of respiration, of course. The hypoxic patient, the hypoxic acidotic patient for that matter, tend to have deep breathing. So we've kind of briefly touched on Kussmaul respirations, and you could look at Kussmaul respirations, that deep, heavy, deep rapid breathing. They are attempting to counterbalance their lactic acid by getting rid of extra carbon dioxide. If they blow off that extra carbon dioxide, they can sort of try to balance out the lactic acid. In many cases, the longer they go, the less successful in doing that they are. So the CO2 is an acid itself, and it can create some acidotic properties for something called respiratory acidosis. So in many cases, just by getting rid of extra CO2, we can greatly improve the body's pH. Although if it's from a metabolic cause, such as, say, a kidney failure, it doesn't. It really has a very difficult time standing up. So, whoops. Okay. So we talked about membrane potential and the process of exchanging the various electrolytes across there. So we're going to move on now to cellular structure and function. So here we have an example of a basic animal cell with a cell membrane. It has a Golgi body in there, rough endoplasmic reticulum, cytoplasm, lysosomes. There's some free ribosomes running around in there, some centrioles. There's a nuclear envelope, the nucleolus within the nucleus itself. We've got some chromatin, mitochondria, smooth and rough endoplasmic reticulum, nuclear pores, ribosomes, so on and so forth. We've got lots and lots of stuff in there. Cells modify based off of what their intent is, what they're supposed to be doing in the body. So not all cells will have all of these items. Not all cells function the same. In fact, we know that there are 3 major types of muscle tissue, and there are so many different more types of just plain organ tissue. So to look a little bit deeper at this, cell membrane. Cell membrane is this boundary. It is referred to as semi-permeable, so it selectively allows some things in and out, and usually that's through the protein gates that are on it. Cytoplasm, or sometimes referred to as the cytosol. The cytosol is the fluid substance in which the organelles are suspended. So, I mean, if you want to think about throwing a can of fruit cocktail in with some Jell-O and letting it, obviously solidify in the fridge, the Jell-O itself is the cytosol. And if you leave it in there uncovered for too long, you know that you kind of get that little bit thicker, chewier layer of Jell-O right on the top there, and that actually would be more like your cell membrane. There is some cytoskeleton, which it doesn't really show much on the previous slide, but this is kind of some support structures. Basically, it's like very fine ribs or support beams that kind of try to help it keep its shape. The centrioles actually are responsible only for one thing. During cell division, the centrioles will move to opposing ends of the cell, and they will help start to separate out the chromosomes, and those chromosomes then will form another cell that's hopefully identical in most cases. Hopefully, in most cases, identical to what we just had as a normal cell. Occasionally, we obviously will have cells that we don't want in cases of cancer and whatnot. You may have cells that are not necessarily good, and in those cases, they will still tend to give you a carbon copy of the original cell. Ribosomes, these are either free-floating or on the rough endoplasmic reticulum. These are responsible for helping to synthesize certain proteins, and proteins have to do a lot with DNA and RNA. The endoplasmic reticulum, it synthesizes lipids and carbohydrates. It helps to put those to work as part of our fuel, and helps to make them a usable product. Then the rough endoplasmic reticulum is lined with those ribosomes, which helps package up the different proteins. We have the Golgi body, or the Golgi apparatus. This is really kind of a garbage disposal system where it packages up the stuff that shouldn't be in the cell and tries to remove it. So the Golgi bodies are really, they're going to go out there, they're going to envelope something up, and they're going to move it towards the edge so it hopefully can be gotten rid of. Lysosomes, this contains powerful enzymes that help rid the cell of damaged organelles and pathogens, and occasionally are responsible for helping to lyse and destroy the cell itself. The mitochondria is also known as the powerhouse of the cell. It produces the ATP, so that's where we saw a few slides back in the first part of this lecture that we would create ATP. And then the nucleus contains RNA and DNA, and that is the obviously the genetic coding that tells the cell how to work, what to do, gives the body its characteristics, and so on and so forth. So again, a lot of these things are just repeated off of what were on that chart before there. We will have some channels within the cell membrane that helps it allow certain things in, keep certain things out. Also can be involved in movement of certain substances via electrical charge. So if we have certain types of electrolytes on the inside and certain types of electrolytes on the outside, it can help activate and deactivate the cell. So again, I talked to you earlier about the phospholipid bilayer. That phospholipid bilayer is those balloons with two strings, I guess, that back up to each other, and they form that semi-permeable membrane that allows some in and out. The cytoplasm fills the cell. The DNA and RNA. RNA is basically modified DNA. Again, is the kind of the message and the messengers. All right. Now vacuoles. Vacuoles was something that we didn't point out earlier. Vacuoles are kind of like a storage system, almost like those storage bags that you get that you put your stuff in. You'll Ziploc them up, kind of suction the air out of them and store your stuff until another time it's needed. That's kind of like a vacuole. It kind of stores it and transports proteins within the cell. And then, the centrioles, they provide the support and form microtubules and the spindle apparatus of the cell, and again are responsible for cell division. Okay, so some of the proteins on the cell membrane and within the cell have a shape that's a specific match to certain substances such as hormones or drugs. These substances can affect cell's function only when a specific receptor for them is present, and this mechanism works like a lock and key. I'll give you a fantastic example of this. It's the opiate receptors that we have within our body. The opiate receptors will only allow opioids, morphine, toluene, dilaudid, heroin, I think I said heroin, coatings, those sorts of things. Those have a very specific structure to them, and they will only allow those drugs to attach to them. The drug Narcan or naloxone has the same shape, and so therefore, and it is a competitive antagonist, so it will also get in there and it will knock out some of the active molecules of the drug that are attached to that receptor site. So it kind of goes in, knocks them out, and takes over, and it will not allow other substances to come and lock in. So in fact, sometimes they will use a combination drug that is partially an antagonist and partially an agonist, or a drug, so say a combination of a Narcan and narcotic together to gain some different effects off of the pain control medication. So tissues are a group of specialized cells surrounded by a matrix, and those tissues generally will form into really four major types. And those four major types of tissues can be seen on page 162, 163. Epithelials, which tend to be linings, tend to be like skin or the inner layers of a blood vessel. We may have connective tissues, and these are fibrous tissues that are more like really kind of more filler. Fat, for example, which is a adipose tissue, is a connective tissue. Obviously, we have muscle tissue, and muscle tissue is very protein rich, changes in size and length, and allows for general movement in some way, shape, or form. And then finally we have nervous tissue, and nervous tissue is very specialized in transporting messages one way or another. And so what does each of these play in the body? Well, obviously we have to have some linings. We have to have the connective tissue in order to really create a significant amount of our structure, because bones and cartilages, those are all considered connective tissue. The adipose underneath the skin obviously gives us a little bit more shape, gives us storage. We have the nervous tissue obviously to send either signals of good or bad and or move. And then of course the epithelial tissue being the lining, so that's our protective barriers. So whether that be the barrier of the lining of the stomach or the skin, that would be our epithelials. So to revisit this topic again, anatomical terminology and topographic anatomy. We've seen this slide before where we have a few of the planes at least divided up here. Whether we're talking the transverse or horizontal plane cuts us into tops and bottoms. We have the mid-sagittal or the sagittal planes, which divides us into lefts and rights. And then we have the coronal or frontal plane, which divides us into fronts and backs. We know that anterior is towards the front, posterior towards the back, inferior towards the feet, superior towards the front. But all in all, the woman in the picture is standing in the anatomic position. That is the normal position that we like to refer to the body in. So it's facing forward, feet forward, palms forward, and that is referred to as the anatomic position. Again, a recap on the body cavities that we kind of touched on in the midterm chapter. We can have the cranial cavity and the spinal cavity, or sometimes referred together as the cranial spinal cavity. We have the thoracic cavity. Within the thoracic cavity, there's another cavity called the pericardial cavity, in which the heart resides in. So it's a cavity within a cavity. We have the abdominal cavity and the pelvic cavity, sometimes referred to together as the abdominal pelvic cavity. So that's the major cavities of the body where, remember, cavity is a hollow opening in which something resides in. Talked about those. A couple of lines of reference. We use these lines of reference when we discuss various body parts and their relation to others. We may be talking mid-axillary. Well, the axilla, your axilla, is your armpit. So mid-axillary would be midway through the armpit, basically trying to divide you into a perfect front and back half if everything was straightened up in perfect alignment. Mid-clavicular would be midway along the clavicle, which is generally from right at the base of your neck out to your shoulder. So it's almost always about straight north of the nipple if the nipples reside in their typical location. Sometimes nipples migrate, but typically it's about straight north of a nipple, say on a very flat-chested person. And then the abdominal cavity gets divided into 9 regions. You may be familiar with the 4 because 4 is what we talk about at the EMT level, the right upper, left upper, right lower, left lower. But when we get to advanced level medicine, we actually look at it more into the 9 regions. And that's the more appropriate division. You can locate these on 164 in your textbook and how they compare to the quadrants that we normally have used up to now. So basically, you need to know that you have the umbilical region, which is everywhere around your belly button. North of that is epigastric. Below that is hypogastric. So epi is above, hypo is below. You have the hypochondriac regions, and those hypochondriac regions are off to the upper left and right. You have the lumbar regions, which are directly left and right of the belly button. And then you have the iliac regions, like your iliac crests of your hips. You have the right and left ones, those to the lower right and left of the umbilical region. Okay, occasionally we also hear about the body and its internal organs being divided into 2 other major cavities, which would include the cranial spinal cavity, which is referred to as the dorsal cavity. Remember, dorsal meaning back, and the ventral cavity commonly being referred to as a combination of the thoracic, the abdominal, pelvic, and you would also need to include in there the pericardial cavity. All right, so common language is spoken in the fields of medicine and science when describing anatomy and physiology of the human body. Why must you be fluent in this terminology? If you're going to appropriately be able to care for your patients, and then in many cases this is going to be when you are consulting either certain medical resources, maybe the patient has some discharge information, maybe you have some direct medical records, maybe you're conversing with a doctor or a nurse and going to have to transfer a patient to another facility. If they're starting to spout out some of these terms, you need to be able to understand them. So being fluent in medical terminology is critical. I will tell you that it is not something that you pick up overnight. It is usually something that takes a number of years to start picking up on. So before you've heard lots and lots of terms. I encourage you again, like I did in Chapter 7, try to break the word down into parts that you know what it means, and in many cases you can get pretty stinking close to what the actual answer is. Okay, so organs. Organs are composed of tissues that work together to perform a specific function. So a tissue is a specific type of cells that are working in tandem. I'll give you an example of some tissues: epithelial, nervous, and muscle. We have an organ that's very important that we can very easily see those 3 types of tissues right now. If we look at the heart is obviously a muscle, so we have muscle tissue there. It's very specific muscle tissue. It's cardiac muscle tissue, which is very different from all the other muscles. The insides of the vessels that feed the heart, as well as the insides of the chambers of the heart and the layers and linings around the heart are all examples of epithelial tissues. And those epithelial tissues, remember, are linings. And then we also have the electrical part of the heart that creates the stimulus and gives us that EKG. And that is actually more nervous tissue in many cases. Sometimes it's kind of hidden within the muscle tissue, but we have that nervous stimulation that goes on throughout the heart itself as well. So that's how tissues come together to form an organ. And then organs will work together to become a system. So, and like I said, most organs consist of a variety of tissues. And then these organs and tissues carry out a very complex set of functions. Okay, so systems are collections of organs and tissues that interact to do this. What systems provide structure to the body? Well, is this a one and only one answer? It's really not. Offhand, most people probably said the skeletal system. And that is true to a certain extent. It gives us the more rigid form and structure, but it doesn't always give us the full function and the full structure that we use. When you add the muscular system to it, that also gives us a certain amount of structure and obviously gives us much more function. And then what system perfuses the body? That would be the cardiovascular system. So it's the heart and the vessels. And in some respects, you also have to include the blood in there, which is sometimes referred to as its own system itself, the hematologic system. They all work together to perfuse the body. But let's not forget, we also have to have the functioning respiratory system in order for us to be able to move the goods in and out. We have to have a functioning urinary system in order for us to get rid of the solid and the liquid wastes. Okay, so for support, movement, and protection, we generally refer to the skeletal system. And like I said, it interacts with the muscular system as well. The skeletal system is generally considered the bones, the cartilage, the tendons, and the ligaments. These provide the body with structure and support and protection. And the bones are a huge reservoir for calcium. The bone marrow is where new blood cells are created. And then the skeletal muscles will allow for movement. So they work in tandem because the muscles will move generally the long bones, occasionally the other things such as short bones, but to create our gross motor movement. Fine motor movement usually is then through the small, the short, irregular bones. And then typically the adult skeleton has 206 bones that is classified in long, short, or flat. They also will refer to some as irregular bones. Now, a good majority of bones in the infant really are not well ossified, and if you take a look at the skeleton of an infant, you won't really see nice formed bones. You'll see kind of faint shadows of where the bones are going to become. And the biggest reason for this is they haven't calcified. So that's why milk gets pushed so much to elementary school kids and even older kids is that's where the calcium is going to go to create those bones. If you don't, then you have very weak, brittle bones that obviously can't handle the structure and function. So cartilage, tendons, and ligaments. Cartilage is more of a lining, and it covers typically bone ends or other parts of bones where they come into contact or function, which we refer to as articulation, with other bones. So it basically helps create a shock absorber slash lubricant surface that helps the bones move against each other. Tendons. Tendons are places in which muscles then connect into bones. Obviously, their major goal or their major purpose is to help create the movement that we're looking for by attaching a muscle to a bone. Whereas ligaments connect bones to bones, and typically the ligaments are responsible for creating a stability and a limitation in most cases to a lot of different joints. So keep your not all of your joints go in all directions. Some go in only very specific areas or directions, and that's because not only because of the structure of the bone, but the ligaments that help to restrict it. So here's a typical structure of a long bone, and when we talk about long bones, we have a number of parts and pieces that we look at here. And on this bone, we can see where it describes the diaphysis being the shaft of the bone, the epiphysis, which you can see is basically the head of the bone or the articulate area of the bone, the metaphysis, which is basically the transitional period or transitional portion of the bone that goes from the shaft into the head. And there's an epiphyseal plate between those, and that's also referred to as the growth plate. And once that solidifies in late adolescence or early adulthood, that ends the bone from becoming any longer. When kids get fractures through that epiphyseal plate, that is when they can have problems with that bone growing correctly for the remainder of their growth. If we look at the inside of the bone, so we do a cross section of the diaphysis there, we can see various layers of bone. The periosteum is the outer layer, the strong outer coating of the bone. We have the compact bone, which is on the inside of that layer, and that compact bone gives it the strength and the rigidity. Within that, particularly on the epiphysis and the metaphysis, we have cancellous bone, and that cancellous bone is the spongy looking bone. It makes the bones it gives them some strength and structure, but it also makes them so they're not so heavy. And then within all of that, particularly the diaphysis, we have the medullary cavity, and that's where the marrow is. And that marrow also has arteries, nerves, and veins that will operate and run through the various areas of the bone. Note that up on the head of the femur, this is a femur, up on the head of the femur towards the top of the picture, they also are outlining the articular cartilage. That articular cartilage is that lined area in which it comes into contact with another bone. There would be articular cartilage on the other end of the bone, on the south end there, on the top of the, I guess in this case, bottom of the epiphyses. So short bones are comprised of basically wrist and ankle bones. They're usually cube-shaped. Sometimes they are trapezoidal or even have some irregularities that hang off of them. If you have an opportunity to look at a skeleton or pull up a picture of the wrist bones, most of them are fairly cube-shaped, but there are some interesting little variations off of a lot of those. Actually, all of those bones have an individual name. We don't. We refer to them collectively as the carpals, but you have things such as the trapezoid and the rhomboid, cuboid. There's a number of funny, interesting names that go along with them. Flat bones typically are those of like the sternum, the ribs, the scapula, and the pelvis, where they're really more of a sheet of bone than anything. Then we'll also have the long bones that are typical of the extremities. We talked about those various parts there. What gets left out of this book, surprisingly, is when they talk about the irregular bones. And the irregular bones are things like your vertebrae. Your vertebrae don't fit really any of the other categories. They have their own very unique and specific structure. Now, we typically will divide the skeleton into two significant sections, two major sections of the skeleton. We refer to the axial and the appendicular skeleton. I'll draw your attention to your textbook on page 168. And on 168, they have outlined in color the difference between the two skeletons. You have the first picture, the left picture on the left, the taller skeleton there, which looks like the one on the screen here. That picture, everything that's in kind of the gold color is the axial skeleton, or the axis, or the core. Everything that's in gray on that one, or if we look at the smaller picture, everything that's in gold on that one would be the appendicular skeleton, or those are the appendages. That includes their girdle that attaches them to the axial skeleton. So if you look up at the shoulders, you see the clavicles and the scapula, and if you look at the lower part there, you see the pelvic bones are a part of the appendicular skeleton. So the axial skeleton is the straight line at the core of the body, skull, spine, sternum, and ribs. The appendicular skeleton then is the bones of the upper and lower extremities, and the shoulder and the pelvis. So what would the consequences of damage to the articular cartilage that covers the bones where they form joints? So if we have damage to that articular cartilage, then we get bones grating one-on-one. The cartilage is supposed to allow them to move fairly freely against each other, relatively. And if we don't have that, then we have bones grating on each other. It's like when your brakes are going out on your car and you have no more pad left on there, it grinds. And it would be very similar to that in your skeleton. And then what are the consequences of an injury to a ligament? Well, remember, ligaments give restriction to joints. So we may have joints that are moving too loosely, and when we have joints that move too loosely, things can get pinched and trapped, and pinged is what we refer to it as, in a joint that normally wouldn't be able to move, to be caught up in there because of the normal limitations of movement. So you end up having a very unstable joint. So we'll move on to muscles. The muscle tissue is built from cells with very unique proteins referred to as actin and myosin. They're arranged in filaments that contract and relax and allow movement. Now, these I like to think of kind of as being layers of Velcro that kind of sit on each other, and they move back and forth, although they don't terribly rip apart. They usually have kind of that connection there, and those, it would be like if we could make Velcro out of rubber, it would kind of give and take a little, give and take. Eventually it is going to tear if you go too far with it. but those muscle tissues, they kind of arranged in different Z patterns that kind of are interlocking and interlinking and allow for that stretching and contraction. When contraction occurs in response to nervous stimulation, it requires a complex chemical interaction within that muscle. So when a muscle contracts, say when we take a breath and our diaphragm contracts, it goes down, it's going to create a significant amount of a discharge of a neurotransmitter. That neurotransmitter then goes to the other side of the synapse. The synapse is where you get the nervous tissue that basically coming in contact with the muscular tissue, or from nerve to nerve for that matter. The synapse is then stimulated, the other side of it is stimulated to actually do its thing. It passes the same message on down the line via the electrical component of the muscle tissue. So there's a ton of chemical and more or less electrical activity that goes on. To relax a bone, to allow it to go back to its normal positioning, usually it's very, very minimal energy expend there, simply for the fact that it's just the normal relaxation returning to its usual. So we have a couple of different types of muscle. We have the skeletal muscle, which is striated muscle. It attaches to bones and allows for movement, and it is voluntarily controlled. It's referred to striated because it has little bands, these little bands that you can kind of see in it. I don't have a good picture that I can show you there, but it has these little bands that almost match up across the muscle. Smooth muscle is involuntary control. It's within your organs. It's almost always in a tube. So everything from your mouth to your rectum is smooth muscle. It is even the stomach itself is made out of a significant amount of smooth muscle. And a lot of the accessory organs that are involved, say the gallbladder, also have some smooth muscle to them. They're basically under involuntary control. We don't have to think about it. You know, we do have to kind of think about it to get up and walk across the room. We don't have to sit here and think, okay, I just ate lunch, digest. So it is involuntary. Also, the blood vessels will change size from time to time. Actually, they're constantly changing size, but and we don't have to put any conscious effort into that. And then the very specialized cardiac muscle cell. It also is under involuntary control. Its very unique function is it allows that heart to pump blood throughout the body. And the extra cool thing about cardiac muscle, it is the only muscle tissue that actually generates its own electricity, its own stimulus to go. It does rely a little bit on some influence from other parts of the body and from the brain and from the muscle tissue and whatnot, or I'm sorry, the nervous tissue and whatnot. But for the most part, it creates its own electricity and does its own thing. So what's the purpose of the smooth muscle in hollow organs? Actually, is to squeeze things along, something called peristalsis. Peristalsis is where it will start squeezing and kind of move it down. Think about the a snake swallowing something. You watch it slowly move down towards the end of the snake. That's peristalsis. The other part of it is that's particularly for the alimentary tract or the gut. But the smooth muscle also helps it change size as needed. So if we decide we need more blood flow or less blood flow, it allows for the body to do that. We talked about that one. Integumentary system. The integumentary system is the skin, and it has three significant layers to it. Really, two is what is classically the skin, and one commonly gets thrown in. But the epidermis is the upper outer protective layer. The dermis is the living business end of the skin. And then below that is the subcutaneous layer. Subcutaneous is probably not technically skin. It's more of a transition padding between skin and, say, muscle tissue or even bony tissue. But the subcutaneous layer often gets put in there because it really is a feeder layer to the dermis. So the skin creates, it is the largest organ of our body, not within it, but the largest organ of the body. It maintains our body's warmth, protects us from external pathogens, produces oil and sweat to maintain fluid balance. It kind of keeps itself lubricated and self-protected with some of its oils. And again, it is our best defense against external pathogens. So the skin also does a wonderful job of maintaining fluid balance, because if we had no skin and we just had exposed, damp, moist muscle all the time, we would end up drying up. And so by having that layer of skin, it acts as kind of the raincoat that keeps our moisture actually in. There's the dermis, the epidermis, and the subcutaneous epidermis. Significant amount of the epidermal layer is actually dead. Several thin layers of dead skin cells, and a lot of those are containing the pigment melanin, which gives us our skin color, and it helps change our skin color gradually to protect us from the ultraviolet rays. Epidermal cells also have a lot of keratin in them, and keratin kind of fills them up after the living parts of them die out. They fill up with keratin, which is more of a fluid-like, gel-like armor, more or less, that helps give a lot of structure and protection to us. There is the living layer, though, of the epidermis that's just below it, and that's where basically the sensory receptors from the dermal layer contact the outside world of the epidermis. So the epidermis, the dermis is a thicker layer. Like I said, that's where the business is. That's where oil glands, the roots of hair, or what could refer to as the follicle, small muscles like the erector pili. You can see the erector pili. We look back here, the erector pili muscles, right there by this shaft of at the bottom of the hair shaft. The erector pili muscle, that's the muscle that gives you goosebumps. So when your body gets cold, the erector pili muscle raises it up, causes the goosebumps, helps to make you look a little bigger in a sense, because it's supposed to make your hair stand up on end. But so that's what's responsible for that. We also have the blood vessels, both capillaries and some smaller veins and arteries potentially, that are going to feed and nourish that layer. And you have a variety of different glands in there, both sebaceous glands and sweat glands. Sebaceous glands typically are creating oils to lubricate the hair, and then sweat glands also are responsible for giving off extra amounts of water to help cool us. So what are the consequences of substantial damage to the skin, such as extensive burns, disease, or abrasions? We have severe damage to the skin. We can't, A, keep out infection, B, keep in the fluids that we need to keep in, C, we may have a very difficult time maintaining our own body heat. Those being the major ones. Most people who suffer permanent consequences, including death due to burns, usually don't burn to death. Usually what happens is they get an infection after the fact because they can't keep it out, because they don't have their armor. Also, they're highly susceptible to becoming hypothermic as well and dehydrated. So those are very big issues with burn patients. Moving to the respiratory system. Respiration, if you remember, is the process of a gas exchange. It is moving air from one spot to another, and typically that is moving it from the alveolus onto the red blood cells, or from the red blood cells to the end organ. the respiration that occurs at the lungs is referred to as external respiration. It's exposed to the outside world. And that from the red cells to the end organ is that of internal respiration. Now remember, the actual process of physically moving air in and out of the lungs is ventilation. And so we can see an example of ventilation here. Diaphragm comes down, intercostal muscles kind of spread. Some of the other muscles pull the lung rib cage out and upward. Allows for negative pressure to form within the chest cavity. Air gets sucked in. And then as we expire, everything just relaxes back. It makes a smaller space, pushes that extra air out. So our function is to obtain oxygen for cellular metabolism and eliminate that carbon dioxide. We have to have that constant supply of that, right? Constant supply of good stuff in, oxygen, water, sugar, and bad stuff out, such as carbon dioxide, lactic acid, and other waste products that are created. So respiration is gas exchange, carbon dioxide and oxygen. We have internal and external, like we've mentioned. And ventilation requires basic principles of physics. So if we don't have the open airway, we can't move air in and out. If we don't change the pressure within the chest cavity, we don't get air moving in and out. So we're going to start to talk a little bit more about the gases that are involved. The gases are generally measured in what is referred to as partial pressure. You will see it abbreviated as PA. Once in a while you'll see people refer to pulse oximetry. Oh, well, their PaO2 is this. And that's not true. That's incorrect. The PaO2 is measured in blood gases when we will get arterial blood gases on patients. It is also the amount of air or amount of a gas within room air. So for example, they show you 79% nitrogen, 21% oxygen, and 1% trace gases, which if you actually add that up, it's 101%. So usually we say around 78% nitrogen. But if you take the total pressure of air, so we'll say, for example, and this is demonstrated on 174 in your textbook, first column, really that second, third, fourth, second, third, and fourth paragraphs there. It talks about this. So we'll take a barometric pressure of 29.92 inches of mercury, which is the normal pressure of sea level. We don't typically use that in medicine. In medicine, we use 760 millimeters of mercury, or also referred to as torr. Millimeters of mercury is also torr, so 760 torr. And this is at sea level. So here in the Midwest, or in the Great Plains, I know Omaha is roughly 1,000 feet. So there's less air on top of us than there is at sea level. And if we would move it to Denver, there's significantly less air on top of us at Denver than they are here or at sea level. So we're not at 760 torr here in the Midwest or in the plains. We're a little bit less than that. But for demonstration purposes, we're going to use sea level because that's a good standard. So at 760 torr, if we multiply 760 times, we'll say, the 79% that they use in the book, if we multiply that by 0.79, we get 600.4, and that would be the partial pressure of nitrogen in room air at sea level. Or we could take and multiply the 21%, so 0.21 times 760 torr, which gives us 159.6 millimeters of mercury. Roughly 160 millimeters of mercury is the partial pressure of oxygen in room air at sea level. Now, you may be going, what in the heck is the difference? Who cares? Well, because of the laws of physics and the way that our body deals with gases internally, these become very important if we have patients who are involved in diving accidents, people who have gone up high in the mountains and whatnot. This changes because we look at the total package of gases that they have in their body. Remember, you still have that 78 and change percent nitrogen floating around in your body. And nitrogen actually becomes the problem in many cases when we're talking about barotrauma or problems with altitude. We know that gases, just like most of our other electrolytes, will diffuse along a gradient from a higher to a lower pressure. So carbon dioxide carried in the form of bicarbonate is bound to the hemoglobin and dissolved in the plasma. So that's where we find most of our carbon dioxide. Our body will look at that as we take a breath of air in, and we have unexchanged gas arriving at our alveoli. We have now roughly, we'll say 16%. We'll say it's 16% oxygen in the alveoli, 21% oxygen, 16% in the capillary on the red blood cell, 21% in the alveoli. They notice that's different. So even if we did the partial pressure, so we could say 21 times 760, we have 159.6 torr of oxygen in the alveoli, or we have 121.6 torr of oxygen in the capillary that's going by the alveoli. They say, hey, that's not equal. That's not the same. We need to do what we can to make things the same. So there's some exchange that goes on. Some of the oxygen that's in the alveoli moves over into the capillary. Some of the carbon dioxide that's in the capillary moves over into the alveolus. So if we said there was roughly 4 to 5% carbon dioxide in the capillary, there's almost 0% in the alveolus. So a significant amount of that is going to try to jump across because it's going to try to make things the same. It's going to try to raise things up. So there's a big difference there. With that big difference, you're going to see a greater switch. So we know that oxygen returning in the bloodstream, returning to the heart, is low, and that deoxygenated blood gets pumped from the heart to the lungs, from the right side of the heart. And when that PaO2 is higher, more oxygen can be carried by the hemoglobin. So let's think about this a little differently now. We say PaO2. Of room air, 21%. The PaO2, 21% of 60 torr, again, 21% of 760 torr, again, is 159.6 torr. Let's put them on a non-rebreather at 90%. 90% of 760 torr is now 684 torr. So we have a extremely oxygen-enriched environment. The body's going to have an easier time moving oxygen one way and CO2 another. We also will refer to as the oxyhemoglobin dissociation curve, which, wow, that sounds very cool and impressive. But the oxyhemoglobin dissociation curve basically says that the relationship between the PaO2 and the hemoglobin saturation. So when we look at that, so when we have this highly saturated hemoglobin, it's that much easier to give it off. When you have, 90% of your hemoglobin is now saturated. 100% of your hemoglobin is now saturated, and it gets down to to your toe, where hardly any oxygen exists down there. It's that much easier now to say, oh, well, yeah, jump on across. We got to even this out, because remember, we're always looking for that equilibrium. So it moves quite quickly with the higher saturations of O2. Don't think that oxygen doesn't come without side effects, though, because we know that there are certain side effects of oxygen, such as it causes vasoconstriction when given in too high of a dose. Hence the reason why in the olden days we used to just ventilate the crap out of people. We'd bag them 40, 50 times a minute, and a lot of times we wouldn't get them back. And after science has kind of gone through and looked at this, they said, oh, well, maybe part of the problem is that their brain continued to starve for oxygen because they had too much in their body. So it constricted all their blood vessels in their brain, and parts of their brain actually starved when the rest of their body had tons and tons of oxygen running around. So again, to look at the upper and lower airways, remember these are the conduits to move gases in and out, moving all the way down to the alveoli. I don't know we need to spend a ton of time on this because it's something that we've known from EMT class. We've touched on it during the airway chapter. We can look at it again here, briefly. Remember that the upper airway is everything from the epiglottis up. Lower airway is below the epiglottis. And the epiglottis is that flap that closes off the lower airway from the upper airway. So the upper airway, it primarily is responsible for warming and humidifying air. It also filters out some particulate debris. So there actually is a reason why you have those nose hairs. As much as a fashion faux pas as they can be, they certainly do a lot to filter out particulates as they're coming into our respiratory tract. Remember, your nose has a lot of twists and turns in it that help there be a lot of exposure to skin, where the skin can help warm up that air and give off some of its moisture, its mucus, to help so it's not so drying to the lower parts of the airway. Obviously, the mouth and the nose are the common openings. We have the oropharynx, or the cavity behind the mouth, and the nasal cavity, the cavity behind the nose. They'll merge in the back to create the common pharynx, or the laryngopharynx, in which then moves downwards towards the larynx. At the top of the trachea is the epiglottis. And that epiglottis is the safety valve that's supposed to keep food and water out and only allow air to move in and out. Just below the epiglottis, we have the vocal cords within the larynx. The larynx is the voice box, composed of a bunch of cartilage. You have the trachea, or the windpipe, which is below that, partially covered on the outside by the larynx. We have right and left mainstem bronchi, which branch into the right and left lungs. The right mainstem bronchi goes a little bit more down towards the south, where the left mainstem bronchi takes a sharp turn to the outside, because the heart sits there underneath that left mainstem bronchi. So it has to kick out a little bit more. The point in which they split is something called the carina, C-A-R-I-N-A. The mainstem bronchi branch down into the various bronchioles till it gets down to the terminal bronchioles, which then become alveolar ducts, and those ducts lead into the alveoli themselves. We have a respiratory membrane, and that is the gas exchange occurs between the respiratory and the cardiovascular system. That is that thin layer of tissue that separates the alveoli from the capillaries. We also have the various organs and the tissue, or various organs in the cavity lined by something called a pleura. We have parietal pleura and visceral pleura. Visceral pleura covers the outside of the lung. The parietal pleura covers the inside of the chest cavity. There's a very small amount of liquid between them that kind of helps them reduce friction. The various sections of the lungs are referred to as lobes, and you have 3 lobes on the right, 2 lobes on the left. So some of that area gets taken up by the heart sitting over on that side. Sometimes that pleural fluid in there between the 2 layers of the pleura becomes either infected or reduced. If you don't have enough of it, or there's inflammation within there, you get pleurisy. The lungs have a portion, and many organs, including the kidneys, have something referred to as a hilum. And the hilum is the basically central part of that organ where nerves, blood vessels, and other important structures come in and out. So in the kidneys, that would be where the ureters come out and where the capillaries and the veins come in. And that hilum is where bronchus, bronchial arteries and veins, pulmonary arteries and veins, lymphatic vessels enter each lung. Those pulmonary vessels carry blood back and forth to the heart for the purpose of gas exchange. Remember that the pulmonary arteries carry blood from the heart to the lungs and the veins from the lungs to the heart. And the bronchial vessels carry the oxygenated blood from the aorta to the lung and return it to the vena cava. So it's an extra blood supply there. So there are 2 phases of ventilation. There's a respiratory cycle. Usually occurs about once every 3 to 5 seconds. It's typical that we see people breathing somewhere in the 12 to 20 breaths per minute. We have inspiration and we have expiration. Typically expiration takes about twice as long as inspiration. So the inhalation is usually pretty quick, and you give them generally around 2 seconds to exhale for every breath, for every 1 second used to inhale. Pulmonary air volumes are measured by spirometry. We kind of briefly touched on that in the diagnostic tools, where we're looking at peak flow. And we're also talking about concepts such as tidal volume. And tidal volume is the total volume of air inhaled in a typical breath. The average for that is 500 milliliters. So about a pop can and a half, if you will, or half a liter. All right, so you're serving on your squad's rescue team at a local high school football game. Mid-first quarter, the quarterback takes a hard hit and goes down. During your assessment, you note a low level of consciousness, decreased level of consciousness, and that his chest is not rising on inspiration. So what do you think of this? Could be a variety of things that have caused this. Could be a nervous issue, which has knocked out the ability for the patient to breathe well. The altered mental status could be from hypoxia, could be from actually something else, taking a blow to the head, for example. We may also have an issue with the lung tissue. The lung tissue may not be functioning appropriately, so therefore it doesn't expand. Could be a bone problem. The muscle, the bones have been broken and can't expand the chest wall well. All right. Moving on. Okay, so the respiratory and circulatory system, moving to cardiovascular. When we talk about the cardiovascular system, we're typically talking about the heart, the blood vessels, and the blood, although note that occasionally people will also refer to the blood as the hematologic system. And the hematologic system has also to do with the immunology part of it as well. So the white blood cells and how they combat various toxins and antigens in the body. Our cardiovascular system is a closed system. Oxygen gets pumped from the left side of the heart through the arteries to the capillary beds, where its exchange of substances take place. It returns to the heart via the veins, through the vena cava, and then back to the heart. Blood is that tissue that consists of plasma and the formed elements. The average adult has around 5 liters of blood, 5 to 6 liters of blood. Plasma serves as the transport medium for those different proteins, nutrients, elements, chemical messengers, hormones, waste products. If you take a look at 180 in your textbook, they have a drawing of a vial of blood there. You can see that 55% of your blood is typically plasma. So the majority of, when they spin a blood vial down, you'll see a majority of it being plasma. And almost half of it, 45% or so, is the red cells. And then there's that roughly 9–10% that is the remaining elements being platelets and white blood cells. Red blood cells are coated in an iron-containing protein called hemoglobin. The hemoglobin is where the oxygen attaches and is bound for transport. Hemoglobin molecules can attach a ridiculous amount of oxygen to each red blood cell. So we're talking roughly 250 million molecules of oxygen can attach to each hemoglobin group and then be transported throughout the body. There's another blood cell. Well, that's one of the measurements that they will look at when doing blood counts. Is they'll look at the hemoglobin, how much hemoglobin is there, but they also will measure one called hematocrit. And the hematocrit you can see demonstrated in that same picture by the percentage of the formed elements in the blood, obviously mostly being red blood cells, compared to the amount of fluid or plasma in the blood. Lifespan of a red blood cell really is about 120 days. Once they become mature, they have no nucleus. They just kind of float around, do their own thing. There can be a number of proteins found on your red cells as well. And those proteins give you a very specific type of blood. They have these antigens that will cause some antibodies to form and start to attack certain things. So we may also have an Rh factor. So our proteins that we can have A, we can have B, we can have A, B, and O. And then you can have positive or negative Rh factors. These are not the only proteins that are found on the cells, but they're the most common ones and the ones that tend to get the most problems. So when you talk about blood, red blood cells, a lot of times they talk about donors and recipients and whatnot. People who have type O blood, particularly O neg, means they have no surface antigens and they don't have the Rh factor. So they're not going to cause problems in most cases when given to just about anybody. So you'll hear them talk about give them a unit of O neg. Well, if they give them a unit of O negative blood, they're not going to create any reactions because there's not those antigens on the surface. Whereas if they gave them, a person with type A blood gave them type B, their body would react and try to destroy all that blood that's coming into it. It creates a lot of turmoil because that's the wrong type, the wrong kind. People with Rh positive blood will do okay if they receive negative blood, Rh negative blood, because it lacks that protein on there. But a person with negative blood would not necessarily respond well to a person with positive because their body's going to see that, oh, there is something bad or something foreign on that one. So it gets to be very tricky when you talk about giving people blood and doing transfusions. Not something you're going to do in EMS. Paramedics are allowed to do to transport patients with blood transfusions, but it's not something that would be done at the AEMT level and not started in the field. So we're seeing an example here in this picture, and this is such a cool picture because I believe this is probably an electron microscope. This is an example of hemostasis, or the forming of a blood clot. The way that hemostasis occurs: first, we get vasoconstriction, so the vessel starts to shrink, slows down the flow of blood through it. Secondly, platelets are activated by different factors, and the body has different hormones, and the starts to then agglutinate at a certain area of the site of the injury. These platelets will start clumping there and form the plug. And then third, the clotting cascade results in the formation of a stable blood clot, where you start to see things such as red blood cells getting stuck into the mix, as well as these fibrin bands. The fibrin goes in there and kind of ties the whole thing together and gives it some strength and support. The white blood cells, or the leukocytes, red blood cells are called erythrocytes. The white blood cells provide protection from invading antigens, and they will support the development of inflammation that results from injury or infection. Inflammation is a normal process. Generally is not a pleasant process, so that's why we often do things to, steer clear of the symptoms. But it is normal. It's our body's normal reaction to something bad happening. Platelets, these are the small fragments that arise from the breakup of larger cells. Typically red cells get broken down into platelets. They get activated by chemical events in response to the injury, which is going to create what's called a hemostatic response. Hemostat, you'll hear about hemostats. Hemostats are clamps that you can put on a vessel to stop it from bleeding. So when we're looking for hemostasis, we're trying to stop that. So in blood, when we do direct pressure, we're attempting to get to hemostasis for that wound. So, and again, like that previous picture showed, this is a series of problems, or a series of items that happen. Constriction of the blood vessels, agglutination of the various cells, and then them being tied down with the fibrin bands. We can look at the internal anatomy of the heart, and as we look at the heart, we see that we've got obviously the greater vessels. The greater vessels are those that bring blood back and forth to the heart, most commonly considered the vena cava, both inferior and superior, as well as the aorta. And then we also have the pulmonary trunk and the pulmonary tree that feeds blood back and forth to the lungs. We have two top chambers referred to as atria. Those are receiving chambers or reservoirs. And we have two bottom chambers referred to as ventricles. And those ventricles are the actual pumping and working parts of the heart. You can see that the thickness of the muscle in the ventricles is much more significant on the left than it is on the right. Remember, the right ventricle has to pump blood back and forth to the lungs. It's not a very far journey. Where the left ventricle has to pump blood back and forth from the heart to the entire body. Within the heart, within those chambers itself, we have, you can note that there is some roughness to the lining or the endocardium. There's kind of ridges in there that helps the blood constantly be turbulent, moving so it doesn't stagnate and create a clot. There are the papilla muscles. They don't have them outlined there, but those are the little bumps coming up out of the bottom there that attach to the various leaves of the valves. The little cords that go from those little papilla muscles up to the valves are called chordae tendineae. And then you actually have the valves themselves, the mitral valve and the tricuspid valve. Additionally, you have the semilunar valves of the pulmonary trunk and the aorta. Those will operate a little differently. The bicuspid and tricuspid, or tricuspid and mitral valves, those rely on those little papilla muscles to pull on the strings to open the valves. Obviously, the heart's a very muscular pump, circulates our blood through the vessels into our cardiovascular system. It's roughly the size of your fist, maybe slightly larger if you don't clench your fist completely tight. And it's located in the part of your chest called the mediastinum. Mediastinum is the area between the lungs, and it is surrounded by a sac called the pericardium. And that pericardium is that protective layer that goes over around the heart to kind of give it a tough armor coating. And then we have the four chambers that function side by side, really as two pumps